All right, so you're here with us because you really want to understand the important stuff, right? And you want all the crucial details, but without getting lost in the weeds. That's what we're all about here on the Deep Dive. Today, we're diving straight into some really significant new developments in Alzheimer's research. We've read through all the latest findings, and we're ready to break it all down for you.
Yeah, and what we found is actually pretty fascinating. There's a real shift happening in how we think about and understand Alzheimer's disease. So today we're really going to dive deep into the role of this inflammatory molecule called IL-12. There's some really interesting research from the Charité Universitätsmedizin Berlin and the Max Delbruck Center. It was published in Nature Aging, and it's revealing just how crucial this IL-12 might be.
That's right. So our goal today in this deep dive is pretty straightforward. We want you to walk away really understanding exactly how IL-12 is involved in Alzheimer's disease and what that means for future treatments. And, it might surprise you because for so long the focus has been on those amyloid plaques, but inflammation is really emerging as a key driver. Okay, so let's start with the basics. What's the brain's normal defense system? Like, how does the brain protect itself?
Well, the brain has its own set of immune cells, and they're called microglia. Think of them as the brain's own personal security force, right? They're constantly patrolling, monitoring the environment, and their job is to clear out anything that doesn't belong. So that includes microbes, damaged cells, and even those amyloid plaques that we always hear about with Alzheimer's.
So it's like the brain's own little cleanup crew, making sure everything stays tidy. But things change as we age, right? Like, what happens to those microglia over time?
Exactly. As the brain gets older, so do the microglia. Some of them keep doing their job just fine. They remain those great protectors. But others, they start to lose some of their effectiveness. Maybe they aren't as efficient at clearing out the debris. And then here's where things get really interesting. Some microglia actually start to release low levels of these inflammatory molecules. And a really key player in all of this is interleukin-12, or IL-12, as we'll call it.
So the very cells that are supposed to protect the brain can actually start to cause problems by releasing these inflammatory signals. Now, for decades, when we've talked about Alzheimer's, it's almost always been about those amyloid beta and tau protein deposits, right? They've been the main focus, the main story.
You got it. And Professor Heppner has talked about this. He's pointed out that for years, Alzheimer's research really was laser focused on these protein deposits. Inflammation was kind of seen as an afterthought, almost a side effect, something that just happened because those plaques and tangles were forming.
So the plaques were the presumed cause, and inflammation was just the consequence. But Professor Heppner's quote really highlights this big shift. Only recently have we begun to understand that inflammation might be a key factor actually driving the disease itself. That's a huge change in how we're thinking about Alzheimer's, right? So what were some of the first clues that maybe this thinking needed to change?
Right. That's a great question. There's some earlier work that was pointing in this direction. For example, in 2012, Professor Heppner and his team published some research in Nature Medicine. It showed that when they blocked not just IL-12, but also another related molecule called IL-23, it actually reduced some of those Alzheimer's-related changes in mice. So that was a pretty big clue that these inflammatory molecules weren't just innocent bystanders. However, they still had a major question to answer. How are they actually doing this? What was the exact mechanism? That remained a mystery.
So they had this really interesting finding that blocking these molecules was helpful, but they didn't really know why. So to figure out the why, they brought together different areas of expertise, right? Professor Heppner's background is in neuropathology, and Professor Rajewski's is in systems biology. And to get to the bottom of things, they use this technique called single-cell analysis. So what exactly is single-cell analysis?
Yeah, it's a really powerful tool. Single-cell analysis basically allows us to look at the activity of genes within individual cells, thousands of them at a time. So instead of just getting an average from a whole tissue sample, we can actually see which genes are being read and turned into proteins in each specific cell. And this is huge. This allows us to understand just how diverse the responses of different cell types are to all kinds of different stimuli. And you can imagine these single-cell experiments, they generate massive amounts of data, like billions and billions of data points. And that's where AI comes in, right? Those sophisticated AI algorithms are essential for identifying patterns and connections that we as humans would never be able to spot just by looking at that raw data.
So it's kind of like being able to listen to the conversation of each individual person in a really crowded room instead of just hearing the general noise. But applying this to Alzheimer's brains specifically presented some real challenges, right?
Absolutely. Professor Rajewski even said that in aging mouse brains, especially those with Alzheimer's disease, the cells are, quote, so stuck together and tangled, end quote, that trying to separate them physically without either damaging them or changing their properties was almost impossible. And you need intact representative cells if you want to get accurate, reliable data.
So they had this big obstacle of how to actually separate the cells. The researchers had to think outside the box. How'd they end up solving this problem?
Well, they came up with a pretty ingenious workaround. Instead of focusing on trying to isolate the entire cells, they zeroed in on the cell nuclei. The nucleus, remember, is like the control center of the cell. It contains the RNA, which carries the genetic instructions. They developed this new method that allowed them to extract and analyze the RNA from the nuclei of each individual cell. And by doing this, they could look at the RNA in more than 80,000 cell nuclei in their study and get a really detailed picture of exactly which genes were active in different cell types. And just to make sure that what they were seeing in these nuclei was representative of what was happening in the whole brain, they cross-referenced their findings with this amazing resource called the Allen Brain Atlas. It's basically this huge database that contains gene expression data from all different regions of the brain.
That is a really impressive way to get around a difficult problem. So what did they find out when they looked at this detailed cellular picture? What did it tell them about the role of IL-12 in Alzheimer's disease?
Well, the big discovery was this. IL-12, the molecule that we mainly knew for its role in autoimmune diseases like Crohn's disease and rheumatoid arthritis, actually turns out to be a major player in Alzheimer's. And it does this by directly damaging two very important types of brain cells, mature oligodendrocytes and interneurons.
Okay, let's break that down. Oligodendrocytes, what do those do in the brain?
All right, so oligodendrocytes, they're the cells that produce myelin. Think of myelin like the insulation that surrounds nerve fibers, kind of like the plastic coating on an electrical wire, right? And this insulation, this myelin, is absolutely essential for fast, efficient communication between brain cells. So what the researchers found is that IL-12 actually leads to a decrease in the number of oligodendrocytes, and it also messes with the integrity of the myelin that they produce. There's actually an image that goes along with this research that shows this really clearly. You can see that when nerve fibers and oligodendrocytes are exposed to IL-12, there are just fewer of them. And the myelin, the insulation, isn't as good. And of course, this has big implications because this disruption in the brain's communication network directly impacts processing speed and the ability to form new memories, which are like the hallmarks of early Alzheimer's.
Yeah, it makes sense. Less insulation, slower communication, and that definitely contributes to cognitive decline. And you also mentioned interneurons. What do they do?
Right. So interneurons, they're super important for cognition and memory. They're like the regulators of the brain. They fine-tune all that complex communication that's going on between other neurons, the communication that's happening when we're thinking and forming memories. And what the study found is that when IL-12 binds to these receptors on interneurons, it actually kills them. They die. And of course, this is a problem because losing these interneurons really disrupts the brain's ability to process information and form new memories.
So basically, IL-12 is attacking the brain on two fronts, damaging the communication network and wiping out key players in cognitive function. And this leads to this cascade of events, this vicious circle, right?
That's exactly right. You have that initial release of IL-12 by some of those aging microglia. It damages the oligodendrocytes and it kills off interneurons. And then all that cellular damage creates even more debris, which activates the remaining microglia. And those activated microglia can become even more dysfunctional and release even more IL-12. And this just keeps the cycle of inflammation and brain cell damage going. And here's another really important point, this whole ongoing inflammation thing. It also makes it harder for the microglia to clear out those amyloid plaques. So it's like a triple whammy, a multi-pronged attack on brain health.
A self-fueling fire of inflammation and damage. So they saw this happening in cell cultures and mouse models. But how did they go about confirming that this same thing, this same mechanism is happening in human brains?
Well, they did this in a few different ways. First, they showed that blocking IL-12 in their cell cultures and mouse models actually did stop, or at least significantly reduce, these harmful changes. Then they used some high-tech imaging techniques like electron microscopy at the Max Planck Institute to visualize the actual structural damage to the myelin and the reduction in the number of nerve fibers that was caused by IL-12 signaling. And then on top of that, they did something called lipidomics analysis. So lipidomics is basically looking at the different kinds of fats that make up cell membranes. This analysis, which they did at the University of Zurich, allowed them to see how IL-12 was actually changing the structure of the myelin. And what they found was that, yeah, the composition of the myelin was altered when IL-12 was present.
And the most convincing evidence came from actually looking at human brain tissue, right?
You got it.
Yeah.
They looked at autopsy tissue from people who had Alzheimer's disease. Yeah. They found this really strong correlation. The more advanced the disease, the higher the levels of IL-12 in the brain tissue. And to really drive the point home, they exposed human oligodendrocytes in cell cultures to IL-12. And guess what? They saw that human cells were also really susceptible to the damaging effects of IL-12. So all of this evidence from different experiments and from actual human samples really supports their findings about this mechanism.
So this is a huge step forward in our understanding of Alzheimer's. What does this mean for potential treatments? What are the implications?
One really exciting implication is that there are already drugs out there that block the action of IL-12. These are drugs that are currently being used to treat autoimmune diseases. And the researchers are very hopeful that these existing medications could potentially be repurposed for the treatment of Alzheimer's disease.
That is hopeful. The idea that we might already have some treatments available that we can start testing for Alzheimer's. Professor Hepner called IL-12 blockers a new arrow in the quiver, which I love that analogy. But even if these IL-12 blockers already exist, are there any downsides? Are there challenges in using them for Alzheimer's, given how complex this disease is?
You're absolutely right. That's a really important question. It is promising that these drugs exist, but we have to remember that Alzheimer's is a multifaceted disease. There are likely multiple factors at play, so it's unlikely that simply blocking IL-12 is going to be a cure-all, a magic bullet for everyone. Professor Hepner makes a good point when he says that we're probably going to need combination therapies that target different aspects of the disease. But this research does show that at least in some patients, the immune system, and specifically IL-12 signaling, is a really important target. And by controlling this inflammatory pathway, we might be able to slow down the neurodegeneration that happens in Alzheimer's.
And because we can actually measure IL-12 in the blood and in cerebrospinal fluid, it means that there's a possibility of intervening earlier, which, with a disease like Alzheimer's, is absolutely crucial.
I completely agree. If we can detect higher than normal IL-12 levels early on, it could help us identify people who are in the early stages of the disease or even people who are at higher risk. And this would mean that we could potentially intervene earlier with these IL-12 blocking therapies, ideally before there's been too much irreversible brain damage.
So this research is really groundbreaking. And it sounds like the research teams involved are already thinking ahead. They're exploring this new hypothesis that has to do with environmental factors.
Yeah, that's right. They're starting to look into a possible link between the presence of microplastics in the brain and the production of IL-12 by microglia.
Microplastics in the brain potentially causing inflammation. That's a pretty disturbing thought. Professor Rajewski suggested that the microglia might have trouble breaking down and removing these tiny plastic particles, and that this could trigger an inflammatory response, which includes the release of IL-12.
Right now, it's still just a hypothesis. But the basic idea is this. Microglia, they're the cleanup crew for the brain. And if they come across something they can't get rid of, like maybe these microplastics, it could put them into this state of chronic activation. And this would lead to the release of inflammatory signals like IL-12, kind of like a distress signal. It's a really plausible mechanism that connects things in our environment to brain inflammation.
It's a fascinating but also really unsettling area for future research. It makes you think about the impact that our environment could be having on our brains.
Definitely. It really underscores how interconnected our bodies are with our environment and how when that balance is disrupted, there can be serious consequences, especially for the complex ecosystem of our brains.
All right, so let's recap. The key takeaway from this deep dive is that researchers have identified IL-12 as a major player in Alzheimer's disease. And it's not just a byproduct. It's actually actively damaging those crucial brain cells that are involved in communication and cognitive function. And this discovery opens the door for potentially repurposing existing IL-12 blocking drugs as a new treatment strategy.
And this discovery wouldn't have been possible without the collaboration of different scientific fields and without these amazing advanced techniques like single-cell analysis. That's what allowed them to actually see the molecular events happening in individual cells in the brain.
It really highlights the power of scientists working together to solve these complex puzzles. And for you, our listener, we hope you've gained a clear understanding of this important research and how the way we're thinking about Alzheimer's disease is changing.
This research is a reminder that our understanding of Alzheimer's is constantly evolving. And by looking at it from different angles, like the role of the immune system, we can find new ways to potentially treat and even prevent this disease.
So as you go about your day, think about this. If something like microplastics, something that seems so small and insignificant, could trigger this whole cascade of harmful inflammation in the brain, what other things in our modern lives might be subtly impacting our brains? It's a complex question, but this research on IL-12 is a big step forward in understanding this puzzle. Thanks for joining us for this deep dive.